Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you're a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hello, everyone, and welcome to the Genetics Podcast. I'm really excited to be here today with Michelle Werner, the CEO of Alltrna and CEO partner at Flagship Pioneering. Michelle is passionate about advancing genetic medicine, pioneering tRNA therapies that we're going to learn a lot about today, and harnessing the power of biotechnology more broadly to transform patient care and lives through her leadership at Alltrna. We're going to have a great discussion. This is an area of biology that I remember from learning about back in the day, but has come quite a long way in terms of how we can harness it to make some pretty amazing breakthroughs. So we're going to spend some time and go back to Michelle's background, but I wanted to just jump right into Alltrna because I think what you're doing is super interesting. And I just want people to get a sense of where the science is right now and what you all are doing. And then we'll double back to learn a little bit about how you got here. So first of all, thank you. Welcome and appreciate you joining me.
Well, thank you so much, Patrick. I'm honored to have been invited to join you. I'm looking forward to the discussion.
Yeah, and I saw you speak briefly at JP Morgan through part of the flagship event. So I got a sense of your business and the science, and I was really excited about the conversation. And maybe we can start with stop codon diseases. It's the way that I think you all kind of elegantly describe the group, a very large group actually of diseases that you're focused on. I would love if maybe we could start with a couple of examples of what you mean by a stop codon disease and what does that universe of these diseases look like that you're going after?
Sure, absolutely. So yeah, we're very much focused on the world of genetic diseases and starting with stop codon disease. So when we think about genetic diseases, you know that there are like 6,000, maybe 10,000, depending on how you count or who you ask. And these are, and those are the ones that, that stem from a different type of mutation, right? And so many different types of mutations cause these disease, but one type of these mutations, which occurs in about 10% of cases, is a nonsense mutation, where, just for those that are less familiar, that's where the code for an amino acid is instead coded for a stop instead. So you've got a truncated or a shortened protein, or perhaps even an absent protein, depending on where the mutation occurs. Now, of all the 300 million people who have a genetic disease, that would mean about 30 million people have that genetic disease caused by a nonsense mutation. And we would put all of these patients into a category of stop codon disease. So that's the collection of patients whose genetic disease stems from a premature stop codon. So it could be hemophilia, it could be PKU, it could be Duchenne muscular dystrophy, it could be Rett. Any one of these genetic diseases has the chance for there to be that genesis of that disease from a nonsense mutation. And so those would be called stop codon disease patients.
Yeah, and there's a particular group that I've always been interested in. I did my PhD in developmental disorders, and a big fraction of these rare and ultra rare developmental disorders are caused by, premature stop codons in genes that we'd call haploinsufficient. If one copy is not, is not conducive to what we'd call a healthy life. And I think it's also worth dwelling on the fact that some of these 30 million cases that you've mentioned are some of the most devastating diseases that we can imagine, right? And so it's not a random 30 million out of the population. There are a lot of diseases that can be devastating. And I remember during my PhD scratching my head a little bit of how will we treat this really broad and heterogeneous group, because it didn't seem cost effective to say, okay, we're going to design something specific for this particular family that has maybe a small handful of cases around the world. But what I love about what you're doing is you're going upstream of that to target the more the root cause and how to fix this stop codon issue more generally. So maybe you could talk a little bit about tRNA technology more broadly. Where does that come in, and how does this differentiate from some of the other forms of genetic medicine people might be familiar with, like gene therapy, CRISPR editing, and ASOs and other kinds of things that people are used to hearing about?
Yeah, so absolutely. So, one of the great things about reclassifying patients into this bucket of stop codon disease really simplifies that challenge, as you just described, of those 6,000 plus different diseases into a far more tangible and easier to address patient population. But of course, it takes a tool, a universal tool that has the ability to transcend disease and to be able to target patients that have these common mutations across those different diseases. And that's where the tRNA comes in, or transfer RNA. So, what the transfer RNA is, and really this is going back to our biology 101 days, is, it plays a fundamentally important role in that protein translation process, where the tRNA, the anticodon of the tRNA precisely binds to the codon of the mRNA. It understands the instruction for each amino acid, and then finds that corresponding amino acid in the surrounding environment and transfers it to the growing polypeptide chain. So when you think about it, it's the tRNA that actually is the physical link between the mRNA, where these codes are, and the protein itself. So you could never have proteins without tRNAs. Now, in nature, there are no tRNAs that can bind to these premature termination codons. And that's why the translation process terminates too early, and you've got this truncated or absent protein, as we've just discussed. So what we're doing at Alltrna then is engineering these tRNAs that actually can bind to these premature termination codons. It means that we can restore that coding sequence. It transfers that corresponding amino acid, the correct corresponding amino acid, as it should have, as it was intended, to the growing polypeptide chain, and then the result is a full-length functional protein. And so this is the approach that we're taking. Now, why is this approach different from every other genetics approach that exists out there, whether it's gene therapy, gene editing, mRNA, you've mentioned ASOs. So first of all, tRNA is actually the only tool amongst all of those that has the opportunity to transcend disease. Now, and that's because the tRNA is, first of all, it's relatively small. It's about 76 nucleotides in length, and it does the same job for these mutations, no matter which protein is being encoded for, no matter which gene is involved. It actually also doesn't matter where the mutation occurs within that coding sequence. No other modality has the ability to be universal in that nature. And each one of our tRNAs is specific to one of these amino acid stop combinations. And so we can take all of these patients who have these stop codon disease and be able to use relatively few engineered tRNAs in order to be able to address those conditions, rather than what has to occur with all these other modalities is a gene by gene, disease by disease approach. So when you have 6,000 plus of these different conditions, you can imagine only a handful of them are going to get the attentions of various different companies and investments. But for us, it actually doesn't matter if there's one patient or 1,000 patient that has this mutation in a particular disease, that we should be able to capture all of them, and maybe even do so in a universal way with the tRNA technology.
Yeah, it's interesting. You hit the nail on the head with one of the follow-ups I was going to ask, which is how many different tRNA engineered tRNAs do you need? I guess is it 60 something? Is it or is it more than that? How many do you need
It's
to
less.
cover the
It's actually
pairwise? Oh
only
yeah.
19 possible amino acid stop combinations. So that's it. I mean, and that's for precise correction, right? So if you need an arginine, you get an arginine. If you need a glutamine, you get a glutamine. And so only 19 engineered tRNAs, but they are not all distributed equally. So in fact, actually only a handful of them would address the vast majority of patients. And so clearly that's where we're starting our efforts at Alltrna.
Interesting, because I guess with CPGs are a lot more likely to mutate. So in de novo diseases, for example, you get certain transitions between the codon you expect and the termination at a much higher rate. That's really interesting. And I just want to play that back because I think it's a really important point. When you take something like CRISPR or ASOs, you need to design an oligo or a guide RNA or something that is specific to the genomic context. But here the context is really at quite a modular atomic level of the triplet code, and so you've got 19 if you want to be able to do perfect correction, or you could do some kind of fuzzy correction, right? If one amino acid is going to be okay in that position, then and it's not perfect, but it doesn't mess up the binding structure too much, so maybe you can go with it. Have I got that right?
Yes, I mean, I think that's one approach, but what we're doing at Alltrna, at least in the first instance, is precision repair. So the exact amino acid stops are the ones that we're going for, and the most prevalent of those are the ones that we're tackling in that first instance. And so we believe that that precision approach, at least in the initial instance, is what's important in order to be able to make sure that we can generate that clinical proof of concept, which I know is something that everyone's looking forward to.
Yeah. I want to come back to this later because I also really love the way you have the potential to maybe flip clinical trials 90 degrees or 180 degrees, whatever the right rotation is. You can be really genotype based versus phenotype based. But I do want to just start with the preclinical journey that you've gone through. I know that at Flagship, you often spend some time really understanding, does this idea have legs? Where's the science going to take us? What were those couple of moments early on that gave you and gave the team and the investors the confidence that this is something you could have a pretty good impact with? What were you looking for in those couple of years in basic science or stealth mode?
So in that typical Flagship way, we always start these explorations with a what if question. And the what if question that the founding scientific team asked themselves is, what if we could restore the protein translation process by focusing on the tRNA? And that then led to a series of what I will say sort of reason to believe type experiments, where at first we had to can we even chemically synthesize a 76 nucleotide structure that has these chemical modifications on it? That had never been done before at that time. We needed to figure out, can we quantify and measure these tRNAs and actually recognize the engineered tRNAs differently, separately from endogenous tRNAs?
Hmm.
We had to figure out if we could actually deliver them to the site of action, and then very importantly, had to rescue protein. So I'd say we spent about a year or two really focusing on those very fundamental scientific questions in order to be able to decide, does this actually have legs to become the idea of a new modality behind it? And all of those experiments are really what led to the founding of the company in 2020, I'd say. And then we launched and came out of stealth in 2021. I joined the company actually in April of 2022. And so now we're really at this, really exciting time where we're taking those initial sort of reason to believe experiments and really demonstrating it in a much more robust way, in a preclinical way, both in vitro and in vivo experiments, to be able to validate the science.
Yeah. So if we talk about delivery and getting it to the right tissue that you want to act on, what does that look like? Is it a lipid nanoparticle? Is it a virus? You have all sorts of different options. How does that? So you've shown that you can create it. You've shown maybe in an in vitro model that you can make the corrections you expect to see, and it'll be tolerated. How do you then actually get it to the right place to do its thing?
So as with every oligonucleotide, right, delivery is, of course, a fundamental consideration for this approach. We are, at least in our first instance, starting with lipid nanoparticles, as you've mentioned. It's a strategic choice that we're making. And the reason for that is because, obviously LNPs have been now, delivered to millions of patients around the world. They're very well characterized, easily available. We understand what the profiles look like in a clinical capacity. And so when we're thinking about de-risking our initial clinical proof of concept, LNPs make sense as a delivery technology for us to go after first. So we will be looking at rare genetic liver diseases in that first instance as a result. But that being said, this is a modality that we believe has the potential to be transformational across, thousands of different diseases that aren't limited to, let's say, the 400 liver diseases that exist. And so we're looking at exploring different types of delivery technology that might be able to address them. In fact, we've actually already explored in vivo 10 different delivery mechanisms. So we're well on our way to that. And certainly it could be an AAV, but it could be other technologies as well. And in fact, what we're doing with our engineered tRNAs as part of the design process is not just, putting the right sequence of the bases together to make up these tRNAs and optimizing for that, but also optimizing for chemical modifications. And the work that we're doing to understand the design rules of these chemical modifications is also getting us, I'd say, well on our way to understand how we might be able to conjugate these in the futures, maybe add linkers to our engineered tRNAs, which may be able to allow us to explore all different types of novel delivery in the future.
You mentioned roughly 10 types of delivery. What are some of the other major examples that people are thinking about, just for those who are curious a little bit more about the
Yeah, so I mean, there
explosion of
are
technology?
AAVs, right,
Yeah.
that you mentioned. There are, conjugations that we've already talked about. There are not just lipid nanoparticles, but other types of nanoparticles that are novel in nature that are being explored. So, I actually feel like this is a science that is really emerging like right
Yeah.
now. And there's so much emphasis on it within the industry, which I think is super exciting. So I do believe that over the course of the next 5 or 10 years, we're going to see a number of these different novel technologies that will be able to help us unlock, the exploration of oligonucleotides in many different capacities in the future, which is super cool.
Yeah, definitely. Is there a part of biology that is still terra incognita we can't get gene therapies to, or is there a delivery method for almost every part of the body, and you'd have to mix and match what you choose?
Well, I believe that there will be a delivery method for every part of the body, but I know the CNS is kind of like that sort of area that is always really tricky because, of course, you've got to think about the blood-brain barrier and how to cross that, or are you going to do, intracranial delivery to try to avoid that? So I'd say that's probably the biggest area that I feel if we can unlock, it really can open up a whole world of possibilities. And the good news is that there is work being done in this space. So will I say that it's impossible? Absolutely not. I don't think anything's impossible. We just haven't figured it out perfectly yet.
That's right. On that similar thread, were there things along the way where there was a timing constraint where you say, okay, we've had this idea for a little while, but actually this has changed and that has changed now, and that makes it possible to do what we do on this kind of scale. If there were any of those, I'd be interested to hear what they were.
Yeah, I always think about Alltrna as, like we stand on the shoulders of giants, right? I mean, the tRNA was identified decades ago, right? But we weren't able to figure out how to unlock it into a new modality until relatively recently. And I think there are a couple of factors at play here. I mean, first of all, even just in the broader world of the RNA space, I mean, you think about what's been happening with mRNAs over the last decade. And then obviously, very importantly, in the last few years, really turning it into a new therapeutic approach. But then also with the work that's been done in siRNAs during this time as well. We've learned a lot from those experiences, how you deliver, how you do chemical modifications, right? What the result is. And so that's certainly something that I'd say we build off of that understanding that has happened before us. But also fundamentally important is that the foundational tools to really unlock tRNA as a new modality didn't exist until relatively recently. In fact, many of those tools had to be developed by us in a proprietary way at Alltrna because they just did not exist. And there's only so many general tools you can use in the
Yeah.
RNA space, but really
Mhm.
nobody had ever quantified a tRNA before. And I think and those tools didn't exist. And I think that's one of the reasons why it couldn't be approached. So I think when you take the learnings and the inspiration from some of these other spaces and then tack on, the sort of single-minded, like understanding of this biology and trying to figure it out and developing some of these tools, this really has been the key to unlocking the science.
Yeah, it's interesting. I'm interested to start to transition to how you all get this into clinical trials and patients. And I alluded to this earlier, but you've got the opportunity to flip the model a little bit. And maybe if we focus on liver disease, an example, you mentioned about 400 genetic diseases, some of them uncommon to common, but some of them rare and ultra rare. But you've got the opportunity to maybe run a basket trial or some kind of other innovative method to tackle this not on a phenotype basis, but on a genotype basis. Maybe you could talk a little bit about how you're thinking about that and maybe explain the concept as well to people who aren't familiar with it, as it's still relatively new. I'm noticing right now your tRNA pin as well on your jacket, which is super. I literally caught my eye and I had to call it out. So yeah, I'd love to
Super
hear about
dedicated,
basket
as you can
trials. It's
see.
great.
Yeah, so I mean, first of all, like the idea, like how we're thinking about our clinical development is something to me that I find super, super exciting. And one that Yeah, anyway, I'm really fascinated about, and one that I actually have some experience with already. So prior to me joining Alltrna, I actually spent much of my career in the oncology space.
Hmm.
And one of the sort of, I'm going to say, relatively common approaches within oncology is to leverage what's called a basket trial, as you described. That's where you have patients across a number of different diagnoses, so different diseases, but they all are unified because they have the same genetic mutation. They all get enrolled in one clinical trial. They get, delivered one therapeutic that is specifically targeted to that medicine, and then you measure a similar or the same outcome across the board, despite the fact that those patients have a different disease. So that's what a basket trial is. It's used, as I mentioned, in the oncology space actually quite prolifically. And this has led to at least 7 or 8 different, I'd say tumor agnostic, but mutation specific type approvals by the FDA. And these types of trials have been ramping up very, very strongly over the course of the last 5 years. So we've seen a huge increase in these numbers of trials in the last 5 to 10 years. And, this is, I think, a play that we can leverage, and tRNA specifically, right, may be amenable to this type of approach because it has that ability to transcend all these different diseases and takes that very mutation-specific consideration. This wouldn't be something that gene therapy or gene editing or mRNA would be able to leverage, but a tRNA certainly could. And so, as we mentioned, we're planning to enter into rare genetic liver diseases. And so our approach is to basket a number of different diseases together. This could be somewhere between, 2 or 3 different diseases, but could be up to 20 or so different diseases into one clinical trial. And they would all be given the same engineered tRNA formulated with a lipid nanoparticle. So what's great about this approach, or very different about this approach, is number one, you can have rapid indication expansion potential that's unlike any other modality because, as you know, instead of doing one clinical trial in one disease leading to one indication, you could get many indications simultaneously, or might even be able to get that disease agnostic but mutation specific indication quite quickly. But then also, when you think about the investment that is required to run all these clinical trials, think about the efficiencies that can be realized by doing one study instead of many across all those different diseases. And that, I think, is a huge differentiator. Not to mention, and I think this is one of the things that is most motivating to me at all, the opportunity to bring in diseases that would otherwise not have a clinical trial at all, right? Think about the ultra rare, the nano rare diagnoses. You would just never do an individual clinical trial for any of those specific diseases if you had to do one disease at a time. This allows us to capture all of those patients and hopefully be able to give them an opportunity that will benefit them, which, might not happen with any other type of approach out there. And so I'm truly, truly excited about the potential to tap into those patient populations and give them something that might transform or alter the outcomes for those patients.
Yeah, I couldn't agree more. I was going to say the exact same thing. You hit the nail on the head that there's this group of people at the very rare end of the spectrum that just get burned over and over by not, that starts with a hopeful preclinical project, but then money becomes a problem or feasibility becomes a problem. And that's also what I really like about this approach. I had maybe a more technical question, which is, I think it's really interesting that your oncology background allows you to come into this new field with fresh eyes, because it's always been a question I've wondered about too, where it's so common then in the oncology field, but much less so in other genetic disease. And I think you answered why, which is if you have a KRAS mutation, it shows up across lots of different kinds of tumors, but you don't really have the same thing where an APOL1 kidney disease mutation kind of only shows up in APOL1 kidney disease and maybe a few others. But if you go up a level to the Sstop codon level, then actually you do have this really kind of elegant umbrella approach. And I was curious about how you measure efficacy in that kind of trial. Do you end up what's the bar you need to cross, for example, to have a label that says this is for every stop codon of this type, or this is for these diseases in this type, because you kind of have a stratification challenge, I guess, right? Where if you only get a couple of patients in the really rare end of the spectrum, then it may be difficult statistically to understand. Is there some kind of grouping, or how does that work to actually determine what the label looks like at the end when
Yeah.
you're be successful in some but not in others.
Yeah, and this is certainly something that we talk all the time, especially as we're, putting our clinical development plans in place. So first of all, I do not believe that there's going to be a requirement for us to demonstrate clinical proof of concept in each and every one of these diseases. So if we just take liver, for example, I mentioned there are 400 liver diseases. Doing a clinical trial for each one of the, or capturing patients across those each 400, I think is going to be an impossible request. And I don't think that's going to be necessary. In fact, if we just go back to the oncology space to where we've had these tumor agnostic indications, usually in these clinical trials, you have 5, 6 different types of tumors in there. You may have patients that are, more from, let's say, colorectal or lung cancer because they're more prevalent cancers, but very few in, let's say, cervical or testicular or, any one of these more rare types of cancers. Regardless of that, you still get these tumor agnostic type indications because the biology, once you're able to demonstrate the biology and, the biology is the same. These are monogenic diseases generally that we're talking about. And so if you can demonstrate that the modality works in that type of biology, we believe that there's going to be, I'd say, flexibility when it comes in terms of broadening out these indications, especially because many of these patients with these types of indications have no other options. So that's at least our fundamental belief. Now, when it comes to what exactly are we measuring, this, of course, depends a little bit. It depends on whether or not and if, much of this will be finalized in the kind of conversations that we have with health authorities over the course of the next period of time. And that might mean that we limit the numbers of diseases to those that share a biochemical pathway. And then when we share a biochemical pathway, then you can at least measure something that's consistent across the board. But it's also possible that if we broaden across more diseases than that, then we might be able to use what's called a global hypothesis test, which would allow us to basically use individual patient reference as their own sort of baseline, and then the impact of the engineered tRNA in terms of what we're seeing in terms of a biomarker or some kind of phenotype that gets measured as a result. So that's what we anticipate would happen, and we'll have to see where we land based on those conversations.
You're pushing the envelope, I think, with the FDA and other regulators in so many like really positive ways. What are the parts where they say like, yes, we're all for this, and what are the parts where you feel like, oh, it's going to take a little discussion to bring them around to this particular concept or aspect of it?
So first of all, I mean, tRNA is a brand new modality. We fundamentally believe that anytime you introduce a new modality, having conversations with health authorities early and often to kind of bring them along this journey with you is absolutely crucial. And that's the approach that we're taking. Basket trials are being used in the rare genetic disease setting, but certainly not as common as they are in oncology. So we do believe that there will be some flexibility there. And what also helps is that what we're hearing from health authorities, not just the FDA, but also the MHRA and EMA as well, they're looking at the problem with rare genetic diseases too. And their narrative has changed to, we'd like to see more many diseases at a time strategy.
Yeah.
So that to me is, almost imploring the industry, in my opinion, to actually explore these types of approaches like basket trials. So we do think it'll take negotiation, of course, as it always does, but we do think that there'll be flexibility there. Where I definitely don't think there'll be flexibility, and rightfully so, is when it comes to what do we need to demonstrate from a toxicology perspective to show that these are, generally safe, both in a, certainly in a preclinical setting, in, as, robust as an in vivo sort of capacity as we can. Again, we don't think we need to do it in every single disease, but at least to demonstrate the safety tolerability in a couple of different animal models is certainly something that we anticipate being, them being quite firm on. And so we're preparing for that expectation as well.
Yeah, like you said, rightfully so. And this idea that you mentioned, which to me sounded a little bit like a synthetic control arm or digital twin type of approach, where you say, okay, let's suppose you're going to enroll 1,000 people with heterogeneous diseases, but the same kind of core genetic driver. There's maybe a challenge that you have around how do we find a control arm that is, well matched. And so there's an idea that I think is probably gaining traction with regulators around, can we use machine learning, AI, medical records to simulate what an outcome might look like. For many of these rare diseases, we know exactly what's going to happen if someone doesn't get treatment because unfortunately that has been the case for decades, right? So we know life expectancy, we know the natural history. I'm curious on whether you're optimistic about that as a change where we may start to see more and more completely eliminated control arms or maybe reduced using this kind of approach or others.
I think this is going to be an it depends kind of situation. So first of all, by no means do I think there'll be thousands of patients in a clinical trial. We're really thinking in these rare diseases, it's going to be a dozen, a few dozen, something along the lines of that. So very few compared to what we might experience with more common diseases. And yeah, I mean, the reality is that only a handful of these rare diseases actually are truly well characterized. In fact, of the 400 different liver diseases that I mentioned, there are actually 20 that I'd say are pretty well characterized. This, means like we understand the natural history and all of that, right? So in those 20, probably a lot easier for us to do because all of that sort of foundational information exists. The rest of them, not so much. And so what I would say is I do believe that we will move in that direction. In the nearer term, it may be easier to focus where those diseases are better characterized because there's less of an ask, especially with a brand new modality. But once the modality is proven, right, to be safe and effective, I do think that those are things that we will be able to overcome, in time. So maybe not from day one, but maybe after the first one of these molecules gets approved.
Yeah, absolutely. I thought I'd just give a quick shout out to a past episode 105 with Warren Huff, who's the CEO, Reata Pharmaceuticals, that we had a really interesting episode about their partnership with a patient advocacy group that had generated a really nice natural history data set in Friedreich's ataxia. And it was really core to them ultimately getting approval from the FDA to have, I think in that case, a reduced control arm. But it's a really great point that you make actually about the natural history only being available for a really small subset of patients. What does that look like for you all? Is it about partnerships with nonprofits, advocacy groups, academics that want to do this work and you can partner in some way? Have you thought about your own kind of natural history studies, patient finding programs, that sort of thing?
Yes, absolutely. All of the above. And honestly, when it comes to these rare diseases, I think that the entire ecosystem approach is crucial, right? No one company, no one scientist has, everything that it needs in order to be able to successfully bring a new technology forward. So we are talking to patient advocacy groups. We have many ongoing discussions with academic experts. We, obviously we're focused on the tRNA, but we're, doing partnerships when it comes to delivery. We also believe that in the future, it's going to be really important for, larger companies besides Alltrna to help ensure that these, medicines in the future can get broadly distributed across different geographies where it'll be impossible for us to build a footprint in a short period of time, maybe over time, but not at least initially. I mean, these are all things, right? That ecosystem approach is really important. The patient finding initiatives are something that we actually believe that while we're starting to do ourselves, especially for the areas that we believe that we'll be entering into, at least in the first instance, but we are doing it with specific patient advocacy groups that sort of represent that disease because we think that they're the best source of information on exactly where these patients are and how they present and all different types of things. So it would be foolish not to partner with them. But we're also partnering with umbrella organizations like NORD, who, again, for us specifically is really valuable because that's an organization that transcends disease. And the work that we're doing from a patient finding perspective, from a genetic mutation, understanding perspective, is something that can be applied across all these different diseases. And we're starting to lay the foundation for that.
Yeah, definitely. How do you think about this, I guess, enviable challenge that a lot of companies, not a lot of companies, but the few companies in your position where you have a really a platform that you can do a lot of different things, versus the money and time that it costs to really do this clinical development properly? How do you think about where you develop in-house, where you partner out? Recently you all have raised 109 million in a Series B, which is obviously a major congratulations and tremendous news for anyone in the rare liver disease space. But you and I know that this only gets you part of the way. How do you think about this enviable challenge of focusing on the golden goose or the egg, so to speak, as you build the company?
Well, you have to do a little bit of both, right? Because the entire value of the company comes from the sort of broad potential of this technology. But we also have to be very responsible with the proceeds from our Series B as we can be. So a couple of things, when we think about engineered tRNAs, we're talking specifically about nonsense mutations, but there's a possibility that it can address missense mutation and frame shift mutation as well. But we have to start somewhere, and generating that initial clinical proof of concept is really crucial. And that's really why we're focusing on the engineered tRNAs in these nonsense mutations, or stop codon disease specifically, and rare liver stop codon disease, right, in that first instance. So it is a little bit about how do we maintain and build some of the capabilities that we know are going to be crucial no matter which direction we take the platform, but at the same time, really double down and triple down in the areas that we believe are going to be fundamental to demonstrating that initial clinical proof of concept, because that clinical proof of concept will be crucial, right, in order for us to be able to secure additional funding so that we can expand to different tissues and to different mutation types, and then maybe even beyond engineered tRNAs. Maybe we want to look at tRNA pools and tRNA fragments. And so that's something, we're not foolish enough to believe we can do all of that from day one, right? But making sure we have, an eye or half an eye focused on the platform, while we're really making sure that we can demonstrate clinical proof of concept as quickly as possible.
I mentioned your background a little bit. You've held a number of positions at great companies, including AstraZeneca and Novartis. What did you learn in large pharma that you've taken with you and it's been really helpful in a biotech? And then on the flip side, what are the things that you felt you needed to unlearn or do differently that just don't translate from large pharma to biotech?
Yeah, so I would say I credit a lot to big pharma for, the 20 plus years that I've spent there, to really giving me a broad understanding of the industry, various different roles across different geographies and different functions. Not only do I understand sort of like drug development, but I also know how to commercialize and launch medicines, and also think about payers very importantly, because at the end of the day, we need to make sure that these are therapeutics that can get reimbursed and ultimately in the hands of patients. So to me, this was such a tremendous foundational experience that, certainly I believe has led me to where I am today, and hopefully will be the kind of critical tools to ensure that Alltrna is successful. Some of the things that I'd say I had to unlearn maybe is, when you do something in a pharma company, a large organization, you've got dozens of teams that you can tap into for expertise. And whilst that's true to a certain degree for Alltrna, because we sit within the Flagship ecosystem and can tap into that, at the end of the day, Alltrna needs to stand on its own. And so we need to make sure that we're building those competencies and those capabilities here. So making sure we think specifically about what are the most important backgrounds and experiences and competencies we need to have on the team is fundamentally important to the work that we do. I'd say I also would have to unlearn, sometimes when you're in big pharma, you do think about, portfolio prioritization, and we do it at Alltrna as well. But portfolio prioritization in a big pharma capacity really does mean that you got to make choices to focus only on the most lucrative business opportunities. And that might mean that you sort of put some of the science to the side, because you need to focus on those types of opportunities in order to be able to get the value for that organization, in that organization broadly. Now, we're focused a lot on value creation at Alltrna, so that's certainly something that I take from that experience here. But we're very, we don't discount, the biology and the science, and really understanding where can we really demonstrate this first and really prove that it has this potential, as well as thinking about the broader opportunity as well. But we look at it from that different perspective.
Yeah, that makes total sense. I was interested to read as well that you're on the board of a nonprofit called Rare Disease Renegades. And I'm just more broadly, I'm interested in what I think is a really complementary but B important movement of more and more nonprofits and other organizations getting involved in this kind of N of one drug development movement. It's another way to address, I think, some of these rare and ultra rare diseases that may not have the commercial potential that you just mentioned as being a critical driver in pharma, but the science is actually there, and we may be able to change some lives. I wonder if you could just talk a little bit about this patient-led research and an N of one type of movement more broadly? Because I think you do have a really interesting front row seat with an organization that's doing some great stuff.
Yeah, I mean, so I would say let's take a little bit of a step back and even why I got into that space or even Alltrna to begin with. And so Rare Disease Renegades is a nonprofit that my husband and I founded. And yes, I sit on the board and he is the executive director of. We started Rare Disease Renegades following the diagnosis of my son with Duchenne muscular dystrophy, which happened nearly 4 years ago now on his 10th birthday back in the sort of height of the COVID-19 pandemic in May of 2020. And so when he was diagnosed with Duchenne, of course, being in, the drug development space, the very first thing that I did was say, okay, which clinical trials are underway? What do I need to understand? What's the emerging technology that's gearing up to address this condition? And, talk about the N of ones. I mean, there were clinical trials underway, but he was actually eligible for 0 of the therapeutic trials that were in existence. And so that was really disheartening. And so we felt like, especially with such a catastrophic diagnosis, we had to do something. We had to really think differently about tackling this problem. And yes, we have started an exploration of these N of ones and gotten connected to people who are advancing some of the science that we've talked about today, genetic science, right? So whether it's gene therapy or ASOs or gene editing technology in these one of N capacities, because nothing is actually being developed for my son's specific mutation. And so that's something that either we wait around for it to come in existence from others, but quite frankly, we don't have time to wait, and neither do millions of patients with these rare diseases. But we, figure that this is something that we really need to try to tackle ourselves. And being connected to the rare disease community and immersed in it, both professionally and personally now for the last several years, to me, it really, like, we're not the only ones doing this. It's happening with dozens and dozens of different families all around the world. And what it's basically proving is that the science exists, the technology exists, the means exist. And if we just put a dedicated effort into making it happen, it can actually happen. And so if it's not going to be on us to make happen, then who is it going to be on? And so that's really the approach that we're taking with Rare Disease Renegades, but other organizations as well who are doing this. But it's also, I'd say, the main motivator of why I'm so passionate about the work that we're doing at Alltrna, because I hope that our approach with engineered tRNAs may mean that all of these families don't have to do this themselves, that we might be able to help address so many of them for them.
Thank you. Thank you for sharing that. If people want to help, to donate, to take part, to help move some of the activities you're doing forward, it's rarediseaserenegades.org. Is there anything else that you'd want to shout out that people can help you all with? Because I'm 100% with you. It's all the ingredients are there, and motivated and passionate families do make an impact. We've seen it time and time again in this field.
I mean,
right?
Well, thank you so much for that, Patrick. And yes, I mean, what I would just say is that any individual patient to do this on their own would cost somewhere between $3 to $5 million. You can imagine that that is not accessible for most families. So, every little bit helps in terms of us being able to take any proceeds or donations and be able to bring them forward for these populations so that they might be able to make a difference for their kids mostly. So thanks
Absolutely.
for that.
Yep, of course. Just to close out here, one of the things I really like to ask people, because it helps me to think a little bit and broaden my horizons, is one company, technology, or idea that you think is really worth everyone watching, learning more about. Only rule is it can't be tRNA therapeutics. We covered that today. So one other thing that's on your mind of, oh, I'm really interested in X because I think in 3, 4, or 5 years, it's really going to be big and I want people to know about it.
Yeah, sure. So I'm paying attention to another, I'd say, sister flagship company that's called Sale Biomedicines. They're focusing on 2 different types of technologies, one of which is an endless RNA, but the other technology is a natural nanoparticle delivery technology, which I think could be super cool and very important for Alltrna. So these are nanoparticles that are derived from nature, and they shuttle molecules into human cells across a number of different tissues, et cetera. So you can imagine if something like this works, that it might be very differentiated from other technologies, that delivery technologies that we have today, different routes of administration, the ability to redose, maybe a different safety profile, ability to tackle different tissues. Could be super cool and something that I'm paying close attention to. So
Yeah.
I would encourage you to look into them if you haven't already.
I haven't. That's super interesting. What is an endless RNA? I can't get over that name. Whoever made that name is deserves a Nobel
Yeah.
Prize. It's very
Think
good.
about it as a circular RNA. It has no end.
Yes.
Yeah.
Okay. I like that. Well, thank you. I really enjoyed this. I learned a lot. I think everybody listening to this will have learned a lot as well. If people want to keep up with Alltrna, your website is just alltrna.com. You got the .com, which is always great. Anything else that you're looking for? I suppose you're hiring and looking for partners. Anything else you want to shout out?
Yeah, always. I mean, I'd say that, there's fundamentally, this is super exciting space. It has the potential to be truly differentiated from every other genetic approach out there. And the ability to tackle so many different diseases is uniquely suited, to the work that we're doing. And for us, I would just say, watch this space. We're going to transform the world. It's early days, but there's so much potential, and we're all really excited to bring it forward.
Amazing. Thank you so much. I really appreciate it.
Thanks, Patrick.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selective Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.